Okay, I'm going to get started by having each of our panelists introduce themselves. And maybe we can start with Alan Moss.
Thank you. I'm Alan Moss. I'm the Chief Scientific Officer of the Crohn's and Colitis Foundation here in the US. And I'm a gastroenterologist by training. And in my prior role, I did some research and clinical work in FMTs in Crohn's disease.
Thank you. Amine?
Hello, Amine Zorgani. I'm also a microbiome scientist, a microbiologist also by training. I'm the founder of the Microbiome Mavericks. It's a podcast. It's posts about the microbiome. It's also consulting for microbiome companies. In my prior role, I was a director of R&D of a skin microbiome companies, developing literally topical microbiome therapies for skin diseases and conditions.
Thank you. Alex?
I'm professor of medicine at the University of Minnesota. I apologize for the background noise here. I direct the Microbiota Therapeutics program here, which has among its missions, it is focused on development of microbiota-based therapeutics. So it's largely a research program, but in our mission, we take care of patients with C. diff infections, but also support a variety of clinical trials ranging from optimizing immunotherapy for cancer to autism to inflammatory bowel disease. I actually am responsible for the term microbiota transplant. I have to say I never liked the F word in that, but other physicians insisted, and it became the unfortunate fecal transplant, which I've tried to fight ever since, unsuccessfully.
Okay, thank you. Marie?
Hi, my name is Marie Joossens. I'm a molecular microbiologist. I'm not a medical doctor, but actually all my scientific work has basically been on Crohn's disease, or at least my thesis PhD work was in Crohn's disease. I started working on that topic and on the microbiota when everybody was fully focused on the genetic part of Crohn's disease. And I was lucky enough to be involved in many scientific studies on fecal transfer, or I should say microbiota transfer. I'll try to do my best from now on, I promise.
Yes, you better call it the proper name or you'll be in trouble with Alex. Okay, let's get started by Alan. Can you just start us off by telling us what the mechanism of action is for fecal transplant? In other words, how does it work?
That's $1 million question. I think Dr. Khoruts and Dr. Joossens will tell you no one's really sure. I think in C. diff, the current understanding is that the C. diff acquires a niche in your microbiome because of antibiotics for the most part, and starts to germinate and replicate and release toxin, and that's what gives people diarrhea. When you do a microbial transplant, you basically squeeze out that niche, whether it's because of outcompeting them for metabolites, outcompeting them for nutrients, just changing the ecology so that C. diff find it much harder to survive in the setting of other microbes introduced, and then you can essentially squeeze them out. They may go back into the spore form and hang around for a while, but they're not actively reproducing and producing toxin. That's certainly my understanding of how it works. I don't know if the other people have different views, but I think that's the overall idea of how it works for C. diff anyway.
Okay, and we'll dig a little bit deeper into that in a bit, but I mean, can you just provide us with some stats on the efficacy and safety rate of C. diff? I mean, C. diff is the only thing that FMT has currently been approved for. And again, just a little bit of a background so that our audience understands how effective it is for that particular disease.
Yeah, absolutely. I mean,
oh, sorry.
Go ahead. Did you say Alan or Amine?
I thought you said Alan, not Amine. Go ahead. Yeah, go ahead, Amine.
No, go ahead. Please,
go ahead.
Alan, continue.
So I think no matter how you do FMT for C. diff, about 90% of people feel better or it doesn't recur in the short term. Now, most of this is observational studies, not randomized controlled trials. in the randomized control trials, it performs a little bit less effective because, there's the barrier is higher for proving efficacy. But in general, whether you do enema, colonoscopy, pills, nasoduodenal infusion, about 90% of people feel better. The C. diff episodes resolve or do not recur in the short term. So I think that's the efficacy overall feeling of how it works or what the success looks like.
Okay. Does anybody have anything to add to that?
I think if I may add on the safety perspective, I mean, today all the trials haven't reported any significant safety issues, at least for the two approved drugs. But we don't have enough data to know, from 10 years from now or 5 years from now, what would happen to those patients who basically received that FMT. So retrospectively, don't know. But as you know, and many of the panelists would say, FMT is not performed since yesterday. There are many clinical trials done on that perspective. So probably if we go and dig and gather all that information, we'll be able to have better data on the safety. But I think that needs more digging and gathering those clinical trials together. So
Okay,
that's an important point. Yeah, go ahead, Alex.
Could add a little bit. So even though it's really been practiced for about a decade on a reasonably large scale, and a lot of data was not collected during that time. Nevertheless, we do have the AGA registry, which is a multi-center registry that tries to capture the outcome data, including long-term outcome data. And OpenBiome did make an attempt to do that as well. They published a paper on over 5,000 patients and really didn't detect much of a safety signal there. They're updating that with 15,000 that they have captured since that last report. When we partnered with OpenBiome, we insisted the registry was a critical component. So there again, we're seeing registry results. We're not really seeing much of a safety signal. There is a spectrum of C. difficile disease. It can be quite complex. Some patients get fulminant C. diff infection, which works by a different mechanism and probably kills the most people. It's related to C. diff. That's a bit different mechanism that Alan described. That appears to be promising as well, though. But those are really sick patients with a 50% chance of dying within 30 days. And so the procedure spares them a surgery and possibly a life.
So just so everybody, our audience is aware, Alex, can you just give an update on the two therapeutics that have received FDA approval for C. diff, and maybe outline what they are, what they can be used for, because I think that they carved out some specific uses.
Right. Both. So yeah, it's a new era for the FDA. They've carved out a new category of a drug. At least in the US, it's considered a drug, and we can dissect that if you like. And the class is called live biotherapeutic products, LBPs. And there are now two drugs that entered the market. One is called Rebiota, which is an enema. It's with Ferring Pharmaceuticals. It's basically homogenized stool delivered as an enema. And the other is a bit more processed product. It is capsules that contain bacterial spores, and that's from Series Health, which is now Nestle. And what they do is they expose the stool to ethanol, which stresses out microbes, kills 2/3 of them, but the ones that survive are shocked into undergoing a sporulation reaction. So they make spores, and the spores survive the ethanol, and they take that spore fraction and put it in capsules. And it appears that the dominant members of the microbiome that are important for C. diff are indeed contained in that spore fraction. Both of these products are indicated for preventing recurrence of C. difficile infection. as Alan described, that infection can really kind of stick to the patient because the organism is able to hide in various ways. Spores is one of them, and it's just very difficult to eradicate. So people generally respond to antibiotics. Once you stop the antibiotics, the symptoms come back. So in order to prevent that relapse, you need to normalize, you need to repair the gut microbiome. So both products basically are attempting to do the same thing, which is to achieve a basic repair of the microbes in the colon.
So, Marie, are these products available now in Europe?
Well, I was in the commission to discuss what we would do in Belgium concerning FMT, if you would consider it as a medicine, or I mean, we were discussing how to treat it. The problem is, of course, that if you treat it like a medicine, that it has to be standardized, and that is something that is hard. So for now, it's still considered like a transplant in Europe, and that's how it has been dealt with. However, here indeed, it's also only approved for C. diff infections. And just from a microbial perspective, if you think about FMT for C. diff infections, C. diff infection, it's actually kind of an opportunistic infection. It's like when the amount of bacteria is down, or if there's in general less bacteria, and as Ellen already said, there is some niches that are available, then patients get infected or prone to infection. And what you do with an FMT is, and actually you directly target the cause of the disease, being that you repopulate the guts, and as such diminish the chance of the C. diff infection to thrive there. So that is quite important, I think.
So basically, everybody around the world, it seems, researchers around the world are in agreement that FMT is definitely effective and fairly safe to treat C. diff. However, if I go to clinicaltrials.gov and type in fecal transplant, there's currently 440 clinical trials that are running for over 30 different types of diseases, ranging from Crohn's and colitis to autism to bipolar, Parkinson's, and even some types of cancers. So does FMT work differently for these different diseases? And if so, why? So maybe, Amine, do you want to start us off with that?
I mean, does FMT work differently? I mean, first we need to understand how FMT works, right? So as Alan had mentioned, we have a mad idea how it works, mostly by increasing diversity, functionality, and some of the, probably competing with other pathogens as well. It depends on the disease you're targeting. So if you're looking for autism, the likelihood of FMT to work the same way as the mechanism action that we know for C. diff is very small, because they're two different diseases. One of them is gut brain axis communication, and the other one is GI disease. So intuitively, as a microbiologist, I would not imagine that the mechanism of action would work similarly. The other thing is that diseases, while they're different, they don't only depend. So I really try to stress that microbiome research is not only microbiome research. I mean, microbes aren't alone in a sphere and there is nothing around them. There is also the genetic background of those people. There is, all the immune system of those people. So diseases, they have different reactions for the immune system. So meaning that FMT may work for one disease because it's probably microbiome driven disease, while probably they will work less for the other one because microbiome would play a role, but not a significant role in that thing. So this is the differences that people should keep in mind. So it's probably a medical solution that it works today for C. diff, which is great, but probably is not the silver bullet that will solve all the diseases out there, right?
Right. Okay.
I could add to that a little bit. Just from, I think, in general, the audience needs to understand a huge difference. In one case, C. diff, we're generally trying to repair a decimated microbiome. It's not that difficult. All you need to do is implant a community of microbes, and they take off. They'll take care of the rest. One dose is sufficient. In most other cases, what you're trying to do And the reason it's decimated is because those patients undergo repeated cycles of highly potent broad spectrum antibiotics to try to control C. diff. Their microbiome is literally being carpet bombed. In other cases, we don't really subject people to that sort of conditioning. And what you're trying to do is, one, you're trying to somehow displace the indigenous microbiota, which is hard to do because after all, they've been already adapted to that particular host, so-called founder effect. And second, you're trying to, as Amine said, you're trying to impart a specific functionality to that microbiome, which is unlike C. diff, where you're just trying to do a basic repair, and any human donor will do, really, even though we impose strict criteria for donor selection, but realistically, anything will work. In all other diseases, you need some kind of functionality that is specific to that disease. As Amine said, for autism, we're looking, for example, for specific metabolites that come from the microbiome that can influence the nervous system. And in our program, we're trying to stratify donors based on those profiles. In ulcerative colitis, it's probably a completely different pathway. I mean, for example, in our group, we're focused on sulfur metabolism for ulcerative colitis. And one can come up with probably another 100 hypotheses of what to look for. And so we don't know when we're going to approach a rate similar to C. diff, whether it's going to be next year or 100 years from now.
Okay.
If I can add on
Yeah.
to that, it's what you see is for C. diff, it's a simple cause and you treat it, but all the other diseases are multifactorial diseases. And I think with all the research that has been done on all these complex diseases, the fact that we saw that microbiota might play a role got everybody very enthusiastic. And it's indeed very important to stratify and see what part of the disease the microbiota might be helpful, or adapting the microbiota might be helpful. But that means that it's not just one cure for all, that we really have to do it very diligently and really look into it to see whether it fits or not.
Okay, so there's a question that's come in, and it's something I think that is kind of important to clarify, is that there's two ways of administering fecal transplants. It's either through an enema, which is the whole product. So basically, Dr. Kortz, you'd said stool mixed with saline, and then there's the freeze-dried capsules. But the FDA approved products that have come up, they're not necessarily whole stool. So they're not the type of fecal transplants that they are doing in clinical trials. They have extracted specific microbes and that they believe are effective in the treatment of C. diff. Is that correct?
Not entirely. So one of the products, which is an enema formulation called Rebiota, that's just homogenized stool. There's no selection there. The other product, which is the spore fraction, it's not that selected. It's still stool. It's the survivors of ethanol treatment. There's still a variation among different donors, exactly what it is. So I think if people think this is some kind of a synthetic consortium of selected microorganisms, it's not that. It's still a donor-derived product. The freeze-dried preparation you referred to, that's the capsules that we use. That's still basically what we're trying to do is capture the entire microbiome. I don't think, and this is my personal view, we're anywhere being smart enough to design a particular, maybe we can stratify particular donors, but it's still, in my mind, the best repair is whatever nature has come up with.
Okay. So then why the need
Just if I would
for-
add maybe Brenda on that one, just for So the FDA approved our stool derived, as Dr. Alex said, but there are companies today working on consortia. Many of them, they're based on Europe as well, and they're also in the States. And these trying to use different ways. Some of them they're using 5 microbes, some they're using 20. I mean, they have different approaches. They're using AI and technologies to actually select those microbes, and they're mostly driven by specific mechanism of action. Literally, they're trying to target specific pathways and so on. Because of the microbiota, it's so complex. The selection of those microbes is even more complex. But none of them had yet succeeded to actually bring that stuff to phase 3 clinical trials.
So why complicate it then? So I look at it in a very simple way. It's like, you don't know what the microbes are that you're trying to replace or to reinstate. You don't know necessarily what you're getting. So why can't you just use whole stool? Why the need to move this to make it a drug as opposed to a transplant of tissue?
Well, if I can take that. So first of all, regardless what it is, in this country, in the US, it's still a drug. And the reason for that is the way the US law is written that's different from European or other places. In the US, anything that is not human, and the key word is human, not human cell tissue that treats, prevents, mitigates disease is called a drug. The FDA has made a determination that microbiota is not human. I tried feebly to protest and say, well, this is not just any microbiota. This is human adapted microbiota. It's not like mouse microbiota, right? Well, whatever. The FDA made its decision. So everything microbiota based is a drug in this country. Whether or not that has practical implications is debatable. I think as far as manufacturing is concerned, the same stringent conditions apply. And where was I going with this? The challenge of why not whole material versus some synthetic, right? And at least part of the reason is the practicality of bringing products to market. You need to raise considerable amount of capital in order to complete the clinical trials and bring a product to market. And the first thing that the investors will ask for is, what is your intellectual property? And the whole stool that's kind of already taken. So you can say, well, I want that microbe and that one and that one, and I think those are the key ones. And you can sell the idea to some investors. At least that's part of the reason, part of the driving mechanism. I'm a little bit cynical here, and I think I'm a bit of a minority view on just how optimistic one is that we can create these consortia from scratch. It's not an idea that a lot of the investors will like to hear. It's also not an idea that a lot of the funding agencies from the government, like NIH, will say that they care to hear about. Science likes to be very reductionist, building from small blocks up. Personally, I think I'm actually with you. I think we do need to do mechanistic research. There is natural variations in microbiome. It's possible to stratify different donors based on functional characteristics in their metagenomics and metabolic capacity. So it's possible to drive into some mechanistic directions from the top down. Probably at some future point, these two directions will meet. Maybe you can spike in some organisms, but really how to build a microbial community and make it stable and resilient, this science is very much in infancy.
Okay, and I want to go back to Amine and Marie and ask you about the status of tissue versus drug in Europe. But Alan, I just wanted you to pop in for a second and give us your opinion on which side of the fence you sit on, because there's two viewpoints on this in the United States is that, you know, it should be allowed to be a transplant. It should be considered a tissue as opposed to a drug which requires going through FDA approval. So what's your personal thoughts on this?
Yeah, I mean, I think I said in the last, my thought is that transplant's a misnomer here. It's probably more analogous to a transfusion than it is a drug or a transplant. It's somewhere in the middle, right? You're altering physiology from cells from a healthy situation to an unhealthy situation. So that might be, but to be honest, as long as the FDA decides it's a drug, really, that's unlikely to change. I think to answer your question on why don't we have just everyone doing fresh donor stool is there's two reasons, right? Scalability. There aren't enough healthy donors around to meet the needs of all these conditions, even if they were all shown to be efficacious for FMT. And the second is safety. There's lots of, to quote Donald Rumsfeld, unknown unknowns in stool, you know, viruses, parasites, metabolites, things that we just can't quantify. And actually COVID, Alex mentioned, was a lesson. We quickly discovered SARS-CoV-2 was transmitted through stool or is detectable in stool. And that was a quick lesson on the importance of viruses that we didn't even realize were there. So I think so scalability and safety are the reasons why we're not using fresh donor stool for all these situations.
Okay. So Amine and Marie, what is the status of stool banks and whole stool versus FDA, well, not FDA approved, I don't know what it's called in Europe, but a drug that's available for C. diff, and what are your thoughts on how it should be handled moving forward?
Well, I know here I know here in Ghent, for example, we have a large stool bank. So samples of fecal samples from healthy donors that are being stored and used in the hospital. There are a lot of startup companies that started indeed with making these consortia, but it is true that it's very hard to mimic the original, and so far we have to acknowledge that we're not capable. One of the reasons is, of course, that the complexity of I usually, if I explain it to my students, I always tell them, like, think of a small community. It's like you're transplanting a whole community, and there is already a bakery and there is already a butcher, and the environment has to be convinced that it's a better bakery and a better butcher, and so on and so on. So it's not just by edit that it will work. But in general, I think safety is a huge issue. You've talked already about things that can be transmitted, but one of the other things that is super important is, for example, antibiotic resistant genes that can be transferred. So it's something that we should not ignore as scientists, or as also not in clinic, that bacteria can exchange their antibiotic resistance mechanisms, and fecal transplant is actually also a very good way to do that. And it's very, very hard at the moment to decide who is a proper donor. I agree there with Alex that there are not enough donors for all the different people that are in need for an FMT. Because if you would think of the best donor, I know we had a case study here of a person who used his kid's stool and did a fecal transfer himself at home, which is like, it might sound as a good idea if you're not in the field, but it's like the worst you can do. Because the infant microbiome is not fully ready yet, so it's not stable, and it was actually, the person came in via ER. So it's very hard to decide. You should have somebody who has not been in contact with antibiotics, has a very diverse stool, and so on and so on. And actually, there are not too many people left that would fulfill these criteria.
If I could just weigh in a little bit. Just to reiterate, because I think it's important for the film audience, for C. diff, a small inoculum is sufficient. So there is really no shortage of donors for that. However, if you're trying to displace somebody's microbiome, like with ulcerative colitis, that requires multiple administrations over and over because you're really trying to force this new community. That's a different ballgame where the numbers come into play. With respect to antibiotic resistance, that's an interesting point, and the FDA is concerned about it, but I'm not sure how well they're thinking it through. For C. diff, you got this antibiotic-induced disease. There's a lot of antibiotic resistance genes floating in that residual microbiome. In general, when one looks at FMT recipients for C. diff, the burden of antibiotic resistance genes goes down because you're introducing a healthier microbiome in general. And it does, in my mind, open the question when one tries to repair that kind of decimated microbiome with a small synthetic consortium of organisms, like some companies are trying to do, you may be able to wrestle down C. diff by bringing in particular function, but it's not going to repair the entire microbiome. And so the patient is going to be left to their own devices to rebuild that microbiome. And it really matters where that patient is. The patient is sitting in the hospital, it's floating with all kinds of dangerous organisms. That's going to be their hospital-acquired microbiome, which is likely to be full of antibiotic resistance genes. So the idea of this holistic repair makes total sense in something like C. diff. When we're talking about other diseases and trying to bring in specific functionalities, it's a much more challenging and different game. And I think other approaches, everything should be on the table and should be studied.
Okay. What's the status in Europe in terms of stool banks, like public stool banks? Are there any that have been established? Or is it just done on a clinic by clinic basis?
It is done on a clinic by clinic basis. To my knowledge, there aren't any big. Probably there are in universities that, as mentioned by Marie, in UGent and many others where these FMTs are being performed, especially also in the UK. There are also the UK Biobank. But today, at least from an industry perspective, there is one company. This is the first, to my knowledge, in Europe to have achieved that. It's called Mat Pharma. They've recently announced the opening of a new facility for FMT production with another partner called Sky Pharma. And these are the first ones basically to actually go into commercially produce in a scale, FMT production. And these aren't targeting C. diff. They're most going into oncology space in a disease called graft versus host when for people who are refusing the engraftments. And they're going also to other clinical aspects of oncology as well.
So, and are they doing clinical research for those diseases, or are they
They're in
actually
phase 3. Yeah, they're in phase 3 for graft versus host, and they're recruiting currently for phase 3 for this specific application, right? But they're also trying to go in, but they're literally focused on oncology, and they're trying to focus into melanoma as well, because there are also some research showing that FMT had positively impacted melanoma. It was a study published, I think, not far long ago. But it wasn't that big a study. So as you can imagine, and just one thing, I've seen this presentation like literally 2 weeks ago in Boston when this company was presenting, and they've mentioned that the same FMT, formulation they're using for their oncology space is the same one they're going to use for melanoma space. And I've literally asked them, why do you think it will work? Because they're literally two different diseases, and at least one is gut skin axis and the other one more of a GI disease. They actually supplied more information saying that from a mechanistic point of view, they think that increasing butyrate producing bacteria through the FMT is what had shown efficacy in that case. And they think that doing the same, and because we know that also in melanoma that butyrate is reduced from microbiota perspective, So sometimes it could be literally completely two different diseases, but they might work with the same mechanism of action, probably increasing certain metabolites, and they seem to have kind of positive hope that it will also work there.
So there's also the Tremont Clinic. I think that's in the UK, is it not? It's the Tremont Clinic in the UK. It's a private clinic that you can go to for FMT. And there's there's various clinics in other countries in the world where people can go and have FMT done and pay for it. Is anybody familiar with that? And does anybody have any thoughts on people that choose to do that?
Well, my basic thought, assuming everything is done in a proper way, which is an assumption, a big message should be, you cannot take a C. diff protocol and apply it to other diseases. It's just different when you try and repair that decimated microbiome. As I said, one inoculum, small inoculum is sufficient. Everything else, just like in the movie, requires these repeated treatments. So that I don't think that's what those clinics are really offering for the most part. Maybe some are kind of extended several infusions and stuff, but this is pretty intense treatment regimens. So that there's that discrepancy.
Yeah,
So-
for these, just on that point, Alex's point, for these chronic conditions, this is not a microbiome reset that's permanent. And so a one-off infusion in a private clinic is not going to fix these chronic diseases, unfortunately.
Right, yeah. So now we look at the model that was in place up until, I think, last year in Australia. And of course, Dr. Borody is featured in the film as well. And up until last year, he was able to treat all kinds of diseases using FMT. It says that he performed over 16,000 in his career. And he was of the opinion that because these chronic diseases required repetitive FMT treatment, and many people lived far away from his clinic, and he was not necessarily in a position to to be finding stool donors. So what his model was is that he would advise the person to recommend a stool donor, probably a relative, and he would do the screening, and then he would provide guidance on having them do it on their own, because it needed to be done every day in order to see any results. So what's wrong with that model?
Well, I suppose, so one thing that the FDA immediately, when we were first approached them, we were the first IND in the country, that is investigational new drug application. It was not feasible with fresh material because the FDA was concerned that the donor you could do some testing, and then you can't really rely on the testing that was done a month ago, that it's still valid today. So they wanted testing done on the donor even after, like several weeks after the donation was done. So that really required ability to preserve the material, which is not something one can do. Now, you could say, well, what are the chances of something like that? So I run a stool bank at the University of Minnesota, and I can tell you, really scary things go through people, and they have no idea they happen. They have no symptoms. The other day, we saw Yersinia come through. In my entire clinical practice, I've never seen a Yersinia infection. And there it was, going through a completely healthy individual without any knowledge that that was happening. We test every single lot of material that goes into patients. So I think the safety concern is real. One always has to balance risk and benefits and everything in medicine. If I have a patient, if the patient is like dying from fulminant C. diff and they have a day to live, well, it's probably totally reasonable to just take random turd and put it in if it's going to save their life. But for the most part, that's not the way you treat most patients.
Okay. Does anybody else have any thoughts on that model that was in place in Australia? And I don't know the stats on, you know, the safety stats on it, but it ran for years and years.
Yeah, for me, it's there's also a moral aspect there because you put a lot of pressure on that healthy donor to stay healthy. I mean, that person has an additional liability towards the one he's supplying to, so to say. I can imagine if it's somebody, it has to be somebody who's close to you, and if he or she gets, like, for example, food poisoning, it might not be that bad for the donor, but it might be very harmful for the recipient. So I can imagine that it puts a lot of, yeah, just from an ethical point of view, that it's also not that easygoing.
Yes, a couple decades back, before this became big, the standard practice, if there was a standard, was that the best donor was your intimate partner, with the assumption that you're already sharing everything bad, and so that would minimize the risk. That's the way I started first at the beginning, and quickly realized that kind of partners people were bringing in, I would not want to share their microbes with the patient. I mean, they had all kinds of diseases of their own, and I already was cognizant that the microbiome plays a role in human physiology on all kinds of levels. And I didn't want to transfer diabetes or atherosclerosis or mental illness or whatever else. So that was the idea that no, we have to have the healthiest donors we can find and standardize that. And
Yes.
and not many patients have that access.
Yeah. However, you know, I think that there was a really good example in the film that somebody brought up, and maybe it wasn't in the film, but maybe it was just a discussion afterwards, is that, you know, the donor has a peanut allergy. Well, you know, the recipient is pooing blood 5 times a day, can't leave the house, seriously, you know, serious anxiety issues. She chose to take the risk of having a peanut allergy, right? Simple. You just don't eat peanuts and you carry an EpiPen. So, you know, that's so Marie, to your point, in terms of putting, you know, pressure on the donor, is it any different when you're pregnant and you're carrying a child? You know, there's certain things you give up because you're carrying a child and you love that child unconditionally. So if you were a donor for somebody that, you know, you loved, I don't really think it would be that much pressure on them to stay healthy for your sake.
Well,
That depends a bit. Pregnancy is 9 months, and you know it's after 1 month, so it's a shorter term, so to say. The thing is just that I think it's we should not underestimate the kind of pressure this might put on people also to be the donor. I mean, it's not everybody is willing to do
I
so.
can probably weigh in a little bit because, you know, we run a donor program, and it is run as a research project of its own. There is an institutional review board that overlooks the entire program. And there is that weight on the donors that, boy, my material is going into all these patients, and I don't want to do something that would compromise that. I think it's important, in my mind, that the donor program is built on a volunteer basis, that there is that understanding, a partnership with the donors. And actually written to the FDA about this. This is something that was not thought through in my mind with the commercial products, because those donors are attracted by money.
Right. Yeah.
They're paid a lot. Rebiota is made in Twin Cities, and so that's where I am, and so we see how it's being done. There are literally posters in the bathroom stalls of bars where people are openly using drugs, recruiting donors, telling them, you can earn $1,600 a month by being a stool donor. We already went through that with blood products. the safest blood supply is volunteer blood supply. I think we might at some point discover the same thing with these. Now, none of us live in a germ-free world. We're all sharing microbes. Maybe the risk is not, that as in blood products, but it is an issue that bothers me. And I think there should be bioethicists that really should weigh in on this field because nobody yet has done so.
Yeah.
I
agree.
on
I
that,
think-
Brenda,
Sorry, go ahead.
try to interrupt you, is I think, and Marie may know better, is that the normal microbiome for me may not be where your microbiome needs to be or what your quote unquote steady state is. And the assumption that my microbiome will be good for you may make sense for C. diff, but for other conditions that involve neuropeptides and stuff, I think it's a big leap to assume that's the case.
Mhm.
Yeah.
I agree. So that's what I said, that depending on what you need from the microbiome, you would need another donor. So I think indeed having a broad range of donors, voluntary donors, I agree with that point as well, would be needed to be able to have the microbiota as a completion or as an add-on therapy for certain disease.
So there's a few
That's one of the
questions.
approaches, if I may add in that just specific point. That's one of the approaches also some of the companies are taking is to actually pool different donors together. And that's what this French company I told you about, they're actually doing for oncology. They're putting different donors, again, because they wouldn't know what is the exact microbiome that these patients are suffering, lacking. So they would pool different donors and they would deliver that. And it seems to have quite a good efficacy.
Yeah, in the film, Dr. Moietti is interviewed, and he did that study, and he had 4 donors, and donor B was the guy. Like, for some reason, his stool was effective for everybody that was part of his study. But the problem is that they didn't know why. So that's what his suggestion was as well, is that maybe we need to look at, blending. I just wanted to ask a few questions from the audience because there's a few questions geared more towards, so we're talking about, replacing unhealthy microbiome, but the questions are geared around keeping your own microbiome healthy. So one of Sandra asks, do you know how someone can have a foodborne infection like listeria? I don't know how to pronounce that. I don't know how to pronounce the other one.
That's good.
And not have any symptoms. It seems like their gut microbiome might play a role in the fact that they don't get sick from the bacteria. Does anybody?
Well, actually, the classic there is C. diff, right? You can carry C. diff spores, not have C. diff infection, but transmit them to someone else. And then in that individual, then they may germinate and produce toxins. So actually, the C. diff model is a perfect example of being healthy, but transmitting something that could be harmful to someone else.
Okay.
As I mentioned in our donor program, yeah, we do notice these bad things go through. Most of them are transients. The donor is put on hold. We keep retesting, and these things eventually go away. The reason they stay not symptomatic is probably multiple factors. Part of it is their immune system, part of the health of their microbiome, and ability to resist those pathogens. The C. diff is a great example because that particular organism is an opportunist. It starts making toxins when the competition is down because of
There is also
antibiotics.
physiology. Physiology also plays a role. The gut microbiome, obviously, the intestine is anaerobic. Some of these pathogens are more aerobic. They like oxygen, so they literally you can see them transition, as Dr. Alice said. And also the pH. The intestine is acidic, and like for instance, if you take listeria, the temperature of the body can actually trigger whether it will be pathogenic or not. I mean, say they can switch virulence factor, which basically makes them pathogenic, depending on the temperature. So you can see that also in lab experiment, right?
So another question we have is the role of particular foods, example, sugar alcohols with damaged microbiome, and the role that the appendix or lack of can have, and FMT and damage mitigation and repair. And that's a question that I've had before, the role of the appendix and how that affects the microbiome. Does anybody?
You know, it's been proposed that the appendix is kind of your little bank account, savings account of microbes. And when there's trouble, it closes off so that when the trouble goes through, it can repopulate. It's like an auto FMT mechanism. Maybe that's true. What's true about the gut in general, you know, that if you think about it, this is where the host, the immune system, interacts the most with the microbial world on a daily basis. So in order to deal with that, it's such an important interface. It has built a lot of redundancy in mechanisms of protection. You try to do research and you try to like knock down some antibodies that are made in the gut. Well, there's another mechanisms that come up and counteract that deficiency, and you don't see anything bad happen. So an appendix may be one of those mechanisms that is protective, but if you lose it, for the most part, you know, for an individual, the risk is low. If you look at population studies, maybe there is a correlation with somewhat increased risk of IBD. But, you know, none of these individual factors can destabilize the system on their own typically.
Okay.
With respect to the food question, I would like to say that it's very dull, the answer there. If you think about every advice you know about diverse eating habits and not too many refined sugars, and that all these things have been linked to a healthy gut microbiota. So basically, enough sleep, sports, no overweight, not too many processed foods. All these things are good for your health, but also for your gut health or your microbiota. So that's a bit, everybody's hoping for spectacular answers there, but I'm afraid that
I generally, you know, counsel patients, they receive their FMT for C. diff infection. Natural, most of them ask, what should I eat now? And so, okay, you've got an inoculum of some microbes. Now you've got to nurture them. Now you have to keep them healthy. So for the most part, that advice is the same across medicine. More plant-based foods, less processed foods, leafy greens, whole grains, vegetables, berries among fruits. Fermented foods tend to have some anti-inflammatory effects, whether that's microbes, probably not, but maybe certain factors that microbes make in the process of fermentation. That's kind of the basic advice. And I think it holds true whether you're a cardiologist or brain scientist or a gut doctor.
and just in
There are
general,
some numbers if I can give you. There is a recent study that published literally, I think 2 weeks ago for Nature Foods. It basically looked into the UK biobank. They looked around more than 460,000 people in the database, and they're trying to look which type of food actually increase your life expectancy on the 40s and the 70s. And in both categories, they found that people who ate what they call it longevity diet, more rich like in nuts and grains and fibers and so on, they actually gave these people on the 40s, they gave them 10 years increase of life expectancy. And on the 70s, they gained 3 years to 4 years life expectancy increase. So this is data, and this is a huge database. There is no doubt that getting more fiber-rich diets can improve your microbiome and hence improve your life, whether you're taking medication or not.
There's a caveat to that, in that, well, first of all, that's an association study, but in general, we're talking about foods that are food for the microbes. That's what fiber is. And that butyrate you mentioned, that's a product of fermentation. Now, the ability to utilize those foods also depends on what kind of microbes you have. It's also been shown that if you just have a diet of ultra processed foods that are kind of pre-digested, you can literally starve your microbiome out of existence, and it's no longer going to be able to be recoverable with a healthy diet. And so that's kind of the important nuance, that diet is also highly individual. Depending on the kind of microbiome that you have, you may have different responses.
So we only have 2 minutes left, and I have one last question for everyone. So I read somewhere that in 2014, they estimated that there was approximately 10,000 do-it-yourself FMTs being performed in the United States. That was back in 2014. I'm sure that number has grown. Could there not be a registry for people that are doing it on their own, so that at least that data is being captured? I think that we are living in an environment right now in which people are reprimanded, told, don't do it on your own. And if you do it on your own, you're on your own. But we know people are doing it. So why are we ignoring that? And instead of taking advantage of the fact that, you know, maybe they should be asked to register and provide data, because that to me seems like a rich source of data. Thoughts?
Well, it's not easy to do registry research and get high quality data. That's one. And, you know, kind of the obvious one problem could be the responders who are volunteers. There's going to be a highly select group of responders. And so you really need to capture everybody if you're going to do a registry to really get a comprehensive look. So it's a huge feasibility challenge.
Amine and Marie, do you have an opinion on that?
Well, I don't know what the numbers in Europe are. I know that Social Security is different, so I think that might play a role as well. But indeed, we see it here as well, that people try to do it on their own. The problem is that we usually only know it when things go wrong. So I think indeed, I'm not a big fan of it, because there are alternative ways to be helped, at least here. But yeah, there are problems with registration. From a scientific point of view, it would be very helpful, but I think we have to be realistic and be aware that it will be only a subset of people who really register then.
I think a better project for the citizenry would be to appeal to the regulators that there should be a nonprofit option for people who can't reach these commercial products because they're priced out or whatever the access problem is. I mean, this is a unique kind of a situation. Nature has developed these products for the most part, and there's no reason why people shouldn't own them.
I mean, last words before we end.
No, I think I definitely join what was just being said. I'm a scientist, and I like when things are clean and I can follow the changes and so on. When you do it on your own, I have no clue how do you did it, when did you did it, what happened, and you don't take. You may be meticulous, but not everybody is. So it's very difficult that I can actually use those data. For us, when you're doing an experiment on 24 hours and you miss one data point in the middle, it's like, oh God, what happened in there? Because I've seen a spike or I've seen something. And that's the problem. So when you don't follow and you don't have all the data, you just can do nothing with them afterwards. Maybe you just have an idea, but I'm not saying that DIY, that the people are doing it because they actually need to do it, is a bad thing for them. I mean, they have no choice. Probably that's why they did it. But these are more aspirational. They are there to inspire companies and regulators to actually act. However, rigorous science is needed for evidence to be able to carry on the journey. So.
Great, thank you. Listen, thank you so much. This has been so informative. We didn't get to all the questions. If we didn't get to your question, please send it to info@designershitdocumentary.com, and we'll try to get one of our panelists to respond to it. I want to remind everyone that Designer Shit is now available to the general public, so you can stream it on Amazon, Apple TV, and Vimeo. You'll see Dr. Quartz on, or you can buy it or rent it off our website. And this is the last week for our free VOD screenings and panel discussions. So tomorrow is the last day. So at noon, we have Amine, and he is moderating a panel on the future of microbiome therapies. It's going to be great. And then at 6 o'clock, we'll be speaking with Dr. Brody and the other stars of the Designer Shit, Saffron and Al, and Charlie and Sky. So You won't want to miss that one. So you can register at designershitdocumentary.com. And I think there's a link in the chat. So thank you, everyone. Thank you, panelists. That was terrific.
Yeah.
Thanks.
Bye.
Take care.
Bye.
Cheers.